what we'd be covering here are some of very basic CC3D functions, but that's going to help us sort of understand um, how cells divide and basically track from which cell they came from. And we would be using dictionaries to sort of like make lists and find out their parent lineage in this case. I don't know of how much you have actually covered cell visualization fields, but that's the other part we'll be covering in this talk as well. Again, just a reminder, the workshop will be again not live stream, but it's being recorded right now. So what are why do we care about basically tracking lineages? And some of the motivation, like I said, was trying to understand where the progeny came from, which is sort of like let us know properties in development and even in like disease control, to sort of understand the whole trajectory of these cells. And like I said, we'd be talking about growth and division. We would be talking about creating cells in Python as well. So if that's something that you have not covered and are interested in, I hope this will be useful for you. And we'd also be visualizing our cells using these visualization fields. Right. So the first sort of like problems of lineage tracing that people have tackled on goes back actually hundreds of years. The pioneering work and actually tunicates, acetians, nematodes have enabled people to understand from how a single cell actually becomes a multicellular organism. And the first measurements that were actually done on these organisms were actually sort of like through direct observation, like people actually sat down and drew and observed cells for hours and days and make fate maps of where these cells would actually end up in the whole body plan. So that was a lot of work, and eventually with the advent of like transplantation techniques or fluorescent dyes, the lineage tracing became a little bit more easier to understand. Some of the most famous fate maps that have been created in zebrafish in the 1990s or in Xenopus, they were all done with the help of fluorescent dyes. But not also in like development, other factors where these lineage maps could be useful are again in the context of tumor growth, because once you know the source from which these cells came from, you can sort of like understand the source of metastasis and so on. So a lot of powerful techniques by trying to trace back these cell lineages. So while we won't be tackling the broader questions of actually modeling the whole body organism, we will try to make small scale simulations today, where we'll start off with a single stem cell, and we will sort of implement growth and division. And what we will do is, as these cells divide, we would sort of track their current identity number and the last identity of the parent where they came from. And when we have all of this information attached to each of these cells, we can sort of construct these like fate maps from which they came from. And then the idea for this session is not just to sort of explore these growth and division properties again, but you could also apply visualization techniques and sort of try to think about if we change any of these initial conditions or the properties of division and growth, how would they actually impact the broad scale simulation? So just as a quick overview of what we're going to do in CC3D. So suppose we have this blue cell. I don't know if you can see my mouse cursor, okay, probably not. So this blue cell on the left-hand side of my corner is what I'm going to consider as my progenitor. Now, as it divides, it's going to give rise to multiple other cells, which could give rise to other cells. In this simulation, for what we're going to work with, we're not going to make any limitations on the number of divisions each cell can undergo, but feel free to add that to your simulations and see what happens and if you see any different fate maps emerging out of your simulations. So in this case, each cell can keep on dividing for as far as at once. So once each cell divides, it's going to make its own lineages. What we will be keeping track of for each cell are their cell ID numbers. So in Compucell, as you generate each of these cell objects, you can create up a attribute, which is the cell ID. This cell ID is basically going to be unique to each cell. And what you can do is, if we keep track of the cell ID number, which basically gave birth to the progenitor cell, you'll be basically able to construct or backtrace basically the fate map of all the parents from which these cells these cells lineage is a part of. So this is what I mean by the examples on the right. So we could basically attach an ID number to each of these cells. And then as the cell keeps on dividing, we will be attaching the previous ID of that cell to the current list so that it's going to have its own sort of like fate map that it's going to collect over time. I hope that makes sense so far, what our goals are to do. Once we can collect all this information of what these cells ID are, we're going to collect them for visualization and plotting. So we're going to, the way that I put out the simulation, so we'll have, I have uploaded four sets of simulations. We will be beginning with 5_3_4_1 as written down at the bottom of the slide. And then as we move over to 2, there will be some Python syntax. So we could, it's up to you on whatever pace you all think is best. But we will start off with making the simulation together for this next set of simulations. But when we move over to 2, we can again go and download the simulation so that it's easier to look at the Python syntax. All right. Okay. So the first couple of steps to do this, we're going to go back to Threaded, and I'm going to put up the first couple of steps that we need to create this simulation. So head over to Threaded if you're still with me and would like to generate this code for constructing a lineage fate map. Go to Threaded, generate a 500 by 500 lattice. And there are 2 things that I changed in the simulation. One is the lattice dimensions, which I've set to 500 by 500. The other thing, which is not underlined here, but also you could set to, which is a higher pixel copy range of 2. Doesn't make a lot of difference, but that's what I have used in the simulations. And the for now, we can click on the rectangular slab, which will generate the rectangular grid of cells for us. But we will actually be removing that code so that we can start off with a single cell and use our Python script for this. The other thing, if people are okay with this step, the other thing we will do is go to the next threaded step, and we're going to add a single cell type called stem. So we'll only have a single cell type, and we'll label it as stem. I'm going to quickly look at the chat. Oh, thanks, Julia. Good. Okay. All right. I don't know if I can access the polls here. So please let me know if you're okay with these steps or are actually if you're okay with these steps, then I should be good to move on. So again, two things has changed in the first threaded step, which I increased the lattice dimensions to 500 by 500, and I changed the pixel copy range to an order of 2, and the rectangular slab is on right now, but we will basically be removing that part to later on create a single cell using the Python, our Python Stepable file. And in the next step, what I did was I added a single cell type, which I've labeled as STEM. So again, we don't need any chemical fields or diffusions for this part. So I'm going to remove it. The other, okay, so I don't know whose microphone is on. Okay, so the other thing which we want to do is sort of select the plugins that we want. So we want contact. We'll be setting up the contact energies in XML. We need the growth anti-mitosis stepables. I don't know if you have seen them already. I believe you must have, but if not, we can talk more if you need any more help with it. For now, make sure that they're on growth and mitosis, and we'll be selecting volume local plex for our simulations because we will be adjusting the volumes in our main Python steppable. And make sure you have your center of mass and cell neighbors on as well. Right. So the first couple of things we're going to do is we're going to head over to your XML script. And what we now want to do is we're going to just have one step that we need to do here, which is getting rid of that uniform initializer code. So if you see the uniform initializer code in this, just get rid of it. And that's the only change we need to make in this file. Ideally, you could also modify the properties here in this uniform initializer, basically by changing the min and max of these values to sort of place your cell in anywhere in the lattice field, and you can change the width and the type that you're attributing to it. I thought we would get some more experience doing it in Python as well. So that's why I have removed this chunk of code from the XML. So steps so far, we went through the two edit steps, and then now we are at the XML file.
I have one quick question, if that's okay.
Yes.
I'm just making
ahead,
sure. We only have the medium, right? Or did we add cell? Did we add a single cell or just the medium?
We did.
Okay.
We added so in this step, we added a new cell type called STEM.
Oh, okay, gotcha. That's what I missed. Okay, I'm all caught up now.
Yeah, and the next couple of steps are okay for people. So the other plugins we added were contact, growth, mitosis, volume local plex, center of mass, and cell neighbors. Okay. And because there are like fewer of us, we can go slow. We don't have to get through everything. So if you want to spend more time doing this yourself, I'm perfectly happy to spend that time. Okay. All right. So this is the next step where we were at. So the threaded generated 3 different files for us: Python file, stepables file, also a Python file, and the XML file. So in the XML file, what we did was we went to the chunk of code that basically initialized our uniform initializer. And what we are going to do now is we're just going to get rid of it so that we can control all aspects of the cell from the main steppable file. Okay. So in the steppable in the XML code, the part that says uniform initializer, that's what we're getting rid of. Right. So next, what we're going to do is we're going to move on to our steppables file. So move out of your XML file, go to the Python steppables file, and this is where we would be making most of our changes. So if you saw your steppable file so far, you would have seen that it created a single class for you probably by the name of constraint initializer stepable. And what we'll be doing is we'll be using this class, this Python stepable, to add a cell. We're going to modify the properties of the cell, and we're going to and then we're going to move on from there, because we're going to add properties of growth and division. Okay, so the first thing that I would recommend us doing is we're going to go to this class stepable, and we're going to identify two variables. We are going to call them an X position and a Y position. And what these are basically trying to find the center of the lattice. So what we want to do is we want to place a cell at the center of the lattice. And this is just another way in which you can create cell objects in CC3D. You can perfectly, very nicely do this with XML, but this is just another demonstration of if you have a particular set of pixels that you want to cell at, how would you create a cell spanning those particular pixels? So right now we haven't done any steps that actually place the cell at those positions. We're just trying to find out what our favorite positions are. So right now what I've done is estimated what the lattice, what the dimensions of the lattice is. And I'm trying to find the center of the lattice to sort of like place that cell in. So my X position is basically 500 divided by 2. My Y position is 500 divided by 2. So that's what those numbers are. And if you notice that in Python syntax, I also have an int placed in front of it. So the lattice, this Compucell lattice, is basically a collection of some integer numbers. So if you want to place some, if you want to associate some numbers to place that objects in, you need to come up with some sort of your integer numbers. So I need to, like, if I divide, for example, 50 by 3, I won't be able to associate a coordinate with that number because the answer is some sort of a rational number. So that's why I did these two steps where I found out the center of my coordinates and I converted it into an integer number. So it's probably more information than you need, but this is a demonstration of how you can pick up pixels on your Compucell lattice. So that's the first step if you want to write these two lines down. It's self.dim.x, which is basically the size of the whole lattice. And if I divide it by 2, that sort of gives me the midpoint of the lattice. Okay. All right, so once we have done this, the next line, what we want to now add is a CC3D cell. So we're going to go to CC3D Python. We're going to go to cell manipulation, and we're going to go to create cell detailed. So CC3D Python, cell manipulation, create cell detailed. So if you basically go through these steps, what it should produce is something like this. This red box that I have highlighted, this is the code that it's going to generate for you. And we're going to replace that code with the positions that we wanted to put our cell in. So I'm going to walk over the steps of this. So have people been able to create the cell in Python? Okay, thank you, Rosie. So thanks, Carly. So you will see that what it generated for us were basically this X and Y positions. And so what we want to do now is that center of that X position and Y position that we had identified where we want to place it, where we wanted to place ourselves, we would be updating that X value to X position. We'd be updating that Y value to Y position. And in this case, we can sort of like say that let my cells have a size of 10. So that's what I'm going to do. I'm going to put cells with a size of 10. And then I'm going to create up a stem cell. So the next line is basically where you're creating that cell object. So new cell will assign a Compucell object to stem cell. And you see that it also has a bracket containing self.stem. So this is the type of cell that it's creating. So new cell basically creates a cell of type stem. And the next line, what you're going to see is self.cell field. Self.cell field is going to basically assign whatever pixels that you have chosen to basically place the cell object in it. So in this case, what it's saying is that it's going from X position of say 250 to 250 plus 10. And because of weird Python syntax, you need to subtract a number to basically make it have 10 units. But all it's basically doing is it's finding the left side of where you want your cell to be placed at. It's finding the right side of where you want to place your cell at, and it's assigning those pixels to the object of stem cell. Okay. And then once we're done with the step, what we're going to do is we're going to set the target volumes, and we're going to set the lambda volumes for the stem cell that we created. So I played around with two values in the file. So for lambda volume, you can set it to 10 actually, and it might make your cells more happy. But yeah, this was another value that I played with. So you can set lambda volume equal to 10 for the simulation. Okay, so recap of the steps we did so far. After we initialized our files, we got rid of the uniform initializer code from our our XML file. We then moved over to the stepables file, where we sort of estimated what positions I want the cell to begin with. And that's what I'm calling as X position and Y position. I had some sort of like a dimension in mind of how big I wanted that cell to be, which I'm labeling as size. And then I'm using a Compucell function called new cell to create up a stem cell. And I'm using cell field to place that cell into the pixels that's being spanned by cell field. And the dimensions of cell field basically begin from X position to X position plus 10. So goes from a left limit of say X position to X position plus 10. And then similarly for Y dimensions, begins at Y position and goes to Y position plus 10. So basically I have some sort of a rectangular area in which I'm placing the cell object in. Right. Do these steps make sense so far? I'll just maybe pause for like 30 seconds or so. If you have any questions. So we are doing all of these changes in that class constraint steppable that I showed. And we're just filling in information that would let us create the cell in Compucell3D. Yeah, please stop me if you have any questions. Okay. All right. So one thing that you can already try at this point is sort of like running that simulation. You can, if you with the volumes that you have set, with the target volumes and the lambda volumes that you have set it, and with all the properties now defined, if you start the simulation, you can already see that the cell is going to start doing things. So if you want to try that, feel free and go ahead. The only thing is that Python automatically will also generate files like a growth steppable or a mitosis steppable. So those properties, since we have not modified them, they're already on. So they would be doing their own code until we have changed them. So if you run the code now, you might have a cell that's basically like running around dividing. But yeah, feel free to do that. Okay, so now that we have actually been able to create a cell and we know what its volume properties are, we now need to basically keep track of some of its attributes. So one of the attributes that we're going to, which is going to be the most useful for us, and the two other attributes are for you to basically utilize and play with. So the attribute is the list that's written at the top of the stem cell.dict list. So that basically is going to contain all the IDs of the cells that have divided. So if you see the picture on the right, what I'm doing is this cell came out of this blue cell. So it had a parent of 1. And then in an updated cell ID, it has the number 2. So its new list that it's going to hold is going to be 1, 2. So as we generate, as we divide and we grow, we're going to keep adding these cell IDs to each cell so that we can keep track of where they came from. At this point in the simulation, however, we only have one cell, so we don't need to worry about any other IDs. So what we're going to doing here is some Python syntax where the dictionary attribute of list is containing the cell's ID. And like I said, each ID is a unique number that's associated with each Compucell cell. So for example, right now in the simulation, all I have is this blue cell, and then this list attribute contains just that ID of 1. The other two attributes, I will explain as we look over them in a little bit, but we're still going to include, which is stem cell.dict, and it has the other attribute of num divisions, which we will use as a way to keep track of the number of times the cell's lineage has divided so far. So for example, if I'm the green, if I'm this middle green cell here, I would have been part of 2 divisions so far, but this green cell all the way to the top, that would have been the third in the line of divisions. All these attributes, when I'm creating them here, are more for you to explore. If at any time you want to look at cells which have only divided a particular number of times, and you can use these dictionary attributes to sort of collect that information. The other advantage of using this dictionary attributes is that you might have a simulation where you have a bunch of different stepables. This dictionary attribute is sort of like universal within the code, because once you attach a dictionary attribute, it sticks to that cell. It's personal to that cell. So any information that you basically want to share within classes, you can just hop on the back of these dictionary attributes and share. And so this list item that I'm creating is going to be accessible to basically a bunch of other stepables that I might create. This color attribute we will actually come to in a moment, and I can explain what you can do with it. All right. So these were the steps that we took with the constraint initializer. So reading out Rosie's questions, she's asking if there's a place in the manual or cheat sheet that states all the information contained in a cell dictionary or cells.cell list that CC3D automatically creates for multicellular model. So you mean all the internal dictionary properties? So there is a cheat sheet that has been uploaded to the Compucell website, which has a lot of these attributes of each of these cells, which necessarily is not in the form of a dictionary, but basically it's accessible on the website on what they are. So for example, lambda volume, target volume, all these properties, which are already inherently present in Compucell, those you can look up, and I believe it should be there on the website. And if we have time at the end of this, I can share that link again. So by step 4, if you have been able to create the cell successfully, modified the target volume and the lambda volume, and you have added dictionary attributes, you should still be able to run it, and it's going to do the same thing. Like I said a few minutes ago, that it's going to just run around dividing. We haven't touched the growth steppable or the mitosis steppable from our side yet, but you should still be able to see cell dividing. So I'm going to just take a 30-second moment to make sure people are on the same page. Can you let me know if the whatever simulation you have created so far is working, or if there are any issues or other doubts?
Yeah, I'm making quick modification. I think I forgot to add I followed all the code. I think I forgot to click add on the cell.
Oh,
And so
okay.
I'm just gonna redo it, but I think, wait, is there a way to not redo it? To just
Um,
add in that?
so do you have the downloaded files by any chance?
Yes, I
Okay, so
think so.
if you just copy the XML file, or even use that file, but if you just copy all the XML syntax um, from that file to whatever this file that you were creating, then that should work too. Or if you want, we can quickly go over all the steps of creating the cell together.
Oh, no, is it, it's numbered the same way, 5.3?
Yeah, 5_3_
For,
a 4_1. I think
okay,
Julio had a suggestion to make as well.
Yes, you forgot to add the cell type. Is that correct?
yes, I typed it in and I forgot to click add.
Yeah, that happens. I think I'm not. I cannot promise. If you go to the XML code uh, and you delete the cell type plugin section, you can then go to the Compucell3D plugins and try to add the cell type plugin again. It's gonna ask you something. I want to redo everything, and then you said yes. And one time I did this and it worked. Hopefully that will be a shortcut. If not, you do what Priyom suggested.
Oh, okay, I do see it. Okay, I think it's gonna work.
Yeah, then I think I have to define which cells you want, and then of course you're gonna. Uh, if there are other plugins later, that depends on which cell types you have. You're gonna redo all of them. It might work. It's worth a try.
Okay. But we can like keep checking in. Sorry, Carly. Yeah, try restarting NanoHub. What I'll do is after I go through steps 5 and 6, I'll take another pause so that people can catch up if they want to. Yeah. So what we're going to try to do is sort of modify our growth and mitosis steppable. So the moment you create a desteppable file, it automatically gives you a suggested growth rate of how you want to basically grow your cells. Oh, and I'm assuming that you have already seen growth steppable. So in CC3D, one of the ways to basically grow a cell is to basically increase its target volume so that you can keep adding more pixels to the cell. And the rate at which you're increasing the target volume could be any rate that you choose. And Compucell automatically will put a value of 1 in the line that I'm highlighting right now. Here we could just choose 0.5 to make the simulation and growth a little slower. So yes, absolutely. So what it means is that in 2 Monte Carlo steps, you're going to add a pixel. So Sharon's question is, I'm surprised target volume is a float. I would have assumed it was an integer since it refers to pixels. So yes, target volume is going to be ultimately an integer. All I'm doing is it's going to add approximately like 1 pixel in 2 Monte Carlo steps instead of adding 1 pixel every Monte Carlo step. Just slowing it down a little bit. So that's the only change we're going to so that's the only change we're going to make in terms of in terms of the growth steppable. So Sharon's another question is, but it's clearly a float as far as Python is concerned. So yes, so it's you can sort of give it a float number. It doesn't it's not going to crash or die on you. It's not going to in fact, in certain simulations that Julia had worked with, setting it to a non-pixel, non-integer target volume actually makes it a little bit more fluidish, if I'm not wrong. But the ultimate target volume that it can actually assign to it is going to be a pixel. It's just a way of controlling the rate at which this is being added. It can read a target volume of say 45.5. But but it's not going to be able to give you a cell surface of 45.5. It's going to basically increase the pixel when it reaches say 46 or something. So that's basically what I'm trying to do.
By adding this half integer value there, what you do, you create a situation where the cell can never reach its target volume. So even though you have a very high lambda volume, lambda constraint, the cell will still not be frozen because you always allow a pixel copy to go above or below the target. So that's a way to increase the volume constraint without freezing the cell, because the cell will always be accepting a pixel to either grow or shrink. So it's a trick you can use in the simulations.
Julia. The other thing that we're going to that we're going to change is so this is as far as growth steppable goes. We're going to make some changes to the mitosis steppable next. So what we're going to assume is that these cells are going to divide when they reach twice their initial volume. So if we started off with say 100 pixels, we're going to let them grow to 200, and then we're going to divide when they reach the 200 pixels. And the way that Mitosis Stepable does it is, it first collects all these cells that have basically crossed that threshold of say 200 in this case. It's creating a list called cells to divide. And once it has the cells it wants to actually divide, it's going to the other loop, which is this one, and it's dividing the cells along a random orientation. Now, Compucell has functionalities where you could actually choose particular axes to divide the cells in. For our purpose, we're just going to pick up any random orientation, which means that the cuts where the division axis occurs are going to be random for our simulation. But you could also be very specific. You can divide like perpendicular to the x-axis, perpendicular to the y-axis, and so on. Just going to check back with people again. So in the stepables file, so we made changes in the constraint initializer. Then we made change in the growth steppable. And then we made a change in the mitosis steppable. So these are the things we have done so far. And we need to do one more thing to the mitosis steppable, which is something of an unfortunate thing, really, because at the moment you call mitosis steppable, what it tries to do is it tries to switch uh, for any dividing cell, it basically tries to switch parent and child. So if you do not have a second cell type, this might cause a problem. So I wonder if Carly, if you were running the simulation before getting rid of this part, I wonder if your simulation was crashing because of this, because this code is inherent in each of the mitosis stepables. And what it was basically trying to do is find a cell type with an index of 2 and basically convert the dividing cell to that type. We are going to remove this piece of code from our steppable, from that update attributes function. So if you have had any issues, this could be a potential reason. So if you want to run the simulation after getting rid of this part, let me know how that goes. And the final thing, which is where I would say the most important part of the code is, so mitosis stepable is going to control our cell division properties. So this is where your parent cell is going to divide. It's going to create up an object called child cell, which is this piece of code right here. And then what this child cell is going to sort of inherit all the properties of the parent cell, which is basically this function here. And when it does, it's automatically also going to inherit the dictionary attributes that we wrote so far. So for example, if your parent cell was this blue cell with a dictionary list of 1, it's going to get copied to the child cell with the dictionary value of 1 as well. But what we want to do is every time it divides, we also want to make sure we add the dividing cells lineage to that list so that we can keep track of our entire line of divisions. So what we're going to do here in that update attribute function, after getting rid of this weird code that Created generates for you, we're going to now access our child cells dictionaries. We're going to use this 3 same attribute that we used called list, numdiv, and color. And we're going to basically update the child cells ID to that list. We're going to now access that number of division attribute. So the number of division basically is the how many times has things divided before you reach that particular cell. So for example, initially for the blue cell, we had a num division of 0, but now as it makes that green cell, we're basically adding that number of divisions by 1. So that green cell has a num division attribute of 1. So does that red cell. And as it makes more children, that number of divisions is going to increase. So this would be a good time to check in with you again on where you're at. Again, if you're good with the constraint initial steppable changes in the growth steppable, we changed the rate at which the cells were basically changing their target volume. In the mitosis steppable, we said that cells should divide when they reach across a target volume of about 200 pixels. And we looked at this function in the mitosis steppable, which is this weird thing that's trying to switch the child cell and parent cell types. So we're going to get rid of that code, and we're going to add these additional attributes for list, numdiv, and color to our child cell as well, so that we can start keeping track of these lineages. And it's not just for this one cell that you're seeing here. As these cells keep dividing, they're just going to keep adding their IDs to these lists, and these lists are going to keep expanding. And so after clearing all of the bad simulation, if we now let the simulation run, we should get an output similar to this. So right now we haven't tracked anything. We have all the information we need to track, but we haven't done the tracking yet. So can people tell me if this is a simulation that you have, that you're getting an output similar to this, or
Can I see the other slide really quick? I
Sure.
forgot one of the last parameters, color. Oh, color.
And even if you forget this one, it's not such a big deal. It's yeah, but yeah, feel free to write it down.
Okay, thank you.
So the most important ones are the list and the num divisions. Make sure the list will make sure that your the new ID with which these cells are created are copied to that existing parent list. The number of divisions will basically tell you how many times along that lineage line have each cell divided, and the color is basically what we'll be using in the later visualization plot. But yeah, even if you don't have that, it's not too bad.
Would you mind
But make
going
sure.
just for a second back to the previous slides? I think I'm
Sure. Sorry, this one you got, or this one?
Yes. Okay, I got that. Thank you.
So is this the simulation we're able to see itself dividing? Basically, you start off with a single stem cell and you're now getting this like blob of cells so far. So Sharon, can you like when they're dividing, can you look in more closely and see if they're dividing along a particular axis orientation? Right. Awesome, Carly. Anybody else with if they're able to get a blob with dividing cells? Also try changing if you set a lambda volume of 50, try changing it to 10. That will make the cells a little bit more happier. They seem to have a long axis pointing from lower left to upper right. That is interesting. I wonder if the division is not random enough. What other parameters? Maybe if you can share a screenshot. I can't see it here, but we can look at it in the Slack channel if that helps. I wouldn't know of any other reason that it caused them to have, okay. So divide cell random orientation should divide randomly, but maybe look in closer as you as these cells are dividing and see if they actually are following a preferred orientation that's making it. So there's a Slack just for everybody. There's also a Slack channel where as part of the Compucell course, it should be in the morning announcements emails, which lets you share images with each other so that we can troubleshoot problems better. Okay, so is this a simulation people are okay with so far? Overall, like, are you able to get cells to divide and grow? Yes, no, maybe.
Now I have like an error. It's telling me that I'm missing a required positional argument for MCS.
Required positional information for MCS. Which does it tell you a line number or
Oh,
which steppable?
it's saying start. The type error says start parentheses, missing one required positional argument. But it doesn't say a line number or which step.
Oh, start function. That doesn't sound like an error that I would just remember at the top of my head. So we have two options at this point. We can either download the next simulation, or we can, when Julio comes back, maybe we can ask him to take a look at it. Either way.
I can wait till he comes back. I think I've been able to follow. I think it's a technical error. I've been able to follow logically though.
We can also take a quick break when at the next exercise, and yeah. Oh, okay. Julia's back if you want to talk to him over chat. Yeah. Okay. So the next couple of sets. So far, what we have done is we have been able to create cells which have sort of divided and which have grown and sort of divided. What we now want to do is, from this image, which is just showing us sort of the cell field, we want to find out which cells have emerged from which particular cells. And to do this, we're going to need use a feature in Compucell called visualization fields. And I don't know if you have heard of this before, but it's a very powerful tool because it allows us to create field-like objects, which we can attach either to the lattice, the Compucell lattice, or either to particular objects, so that we can color cells or even color regions of the lattice according to the values that the field has. So in this particular case, what we want to do is we want to separate our cells as having certain distinct identities, right? So if we can use these visualization fields to sort of interpret or sort of like connect with the cell dictionaries information that we have been collecting so far, then we'll be able to visualize our information. So this part of the code should be in 5_3_4_2. But we'll also like walk through the entire code. So what we're going to do first is now we're going to right click on our steppables file, and we're going to actually say add steppable. So I don't know if you've done this before, but this is also a cool trick if you don't want to basically copy and paste steppable information, but you need to sort of create your own class for doing other specific things. Then you can sort of use this add steppable method. So again, like right click, add steppable, and then you'll see this like tiny bar at the bottom of the screen. So here what we're going to do is we're going to actually click on scalar cell field. And this scalar cell field is going to associate a number with each of the cells, and it's basically going to visualize that output for us. Okay, so if I go through the steps, I right click on the steppables file I have, I say add steppable, I click on scalar cell level, and I say okay. Then it's going to produce me, it also asks you to input a name. So in this case, I have the steppable name that I chose is called coloring cells. You can pick up your own name. And what it's going to automatically do is create up this steppable file. And it's also going to put this information in your Python file as well. So you don't need to do anything else. So what you have now is the coloring cell steppable, and you will see that inside this, this is your scalar field cell level that Compucell has generated for you. And this is what we're going to be modifying next. Okay, so just add a new steppable by right-clicking on your steppables file. And then you have add a steppable name that you want. Go to click on scalar cell level and then hit OK, and it will create this steppable class for you. Okay, so what we're going to do next is we're going to modify. So one thing that we want is this scalar field cell level to sort of interpret what the colors or some scalar value that I'm giving it to draw us a visualization map. And it also means that it should be accessible between different functions that my steppable class has. So even though I'm creating up a cell cell field in this init function, I'm going to associate it with a name. In this case, I've called it self.field_IDs, and I'm going to access this in some other function, say the step function, so that I can basically plot the visualization field as I go along. So in this case, note that I also need to create a right-hand name for this. So this is something that the Compucell player window is going to interpret. So for display purposes, it's going to understand the right-hand side name that you give it. And you can find it, I'll show you, we can talk more on where you can find the cell ID. And then the left-hand side is really just for us because we want to manipulate this object inside the Python script. All right, so what we did now is we went to coloring cell steppables, we generated a new scalar field, and now what we're going to do is we're going to use all of these trees that we have been making so far using these cell lists. We're going to collect that information to sort of draw our lineage tree. So what is our first step going to be? So on the right side, you might see a little bit of a Python code. If this is some of these functions are too new to you, feel free to copy this code either from the demos that I've uploaded, or feel free to write this down as we're talking. And if you have any questions, please let me know. So the basic goal is for all these lineage trees that we have created, we sort of want to understand what is the starting parent that they came from. For example, if this tree, which has made this cell, which came from the starting cell of 1, versus this cell, which came from 1 to 5, the parents of these cells, of all of these cells, would be 1. But for this specific part of the green cells, the starting parent would be 2. Similarly, for these, all the red cells that would keep going so forth would be 3. And for these cells, these orange cells would be 6. If there are new cells that popped out of the stem cell, this blue stem cell, it would be its own lineage again. So we don't want to color everything with 1 because that's the founding father of all of our lineages. But to begin with, we could color things with the stem cell that sort of was given rise out of this progenitor cells. So basically what I mean is we could color all of these cells which has a cell ID of 2 with green, all of these cells which has a cell ID of 3 with red, all of these cells which has a cell ID of 6 with orange, and we can keep going. So even if this cell number with an ID of 5 gave rise to more cells, it would be colored with 2 because this was the original cell it came from. But if we picked 1, then everything would be colored with the same color because it's going. If 1 gave rise to all of the progenitors that we saw. So, so this is one lineage tree that we can draw, you're free to like pick whatever criteria that satisfies you. For example, you could combine it with the num division attribute that we have stored with all of this, and only decide to pick certain cells that have crossed a certain threshold of divisions. You could even look at your simulation output and say that I'm only interested in cell which divided only in this part of the tissue, and then you can modify their X or Y center of masses to sort of collect only those cells to make a lineage out of. So there are multiple options that you can try to construct your own lineage maps. For this one, what we're going to do and what this Python code on the right is helping us is sort of to extract out the first set of dividing cells that came out of this blue stem cell. So what it does is it goes to each and every single cell, looks at their dictionary attribute of list, collects all the numbers that are not equal to 1. For example, if it collected the IDs from this cell, it would be 2 and 5, finds out what that minimum number is, which would be 2, and collects that minimum number in this minimum list. And because multiple cells would have 2 appearing in them twice, it just finds that unique set of combinations. So all it's trying to do is finding the first set of IDs that all this lineage came from, because that's going to be the minimum ID. And again, this is just one set of classification that I have done. You could, easily pick up your own and play around with it. Okay, so that's the part where we have sort of tried to identify how to extract out the cell lineages that we would actually need. And the next step is where we use this unique parent IDs to sort of now tie it with our visualization output. So this is the part. So right after we collected the unique elements, if you're following the downloaded code, right after those lines, what you will see is this piece of code that sort of is where we are doing the actual grouping and the actual coloring. So let me walk you through it. So as of now, we have this unique list that contains all those unique minimum progenitors that came out of the that we collected. We're going to loop over each of them, and we're going to say that yes, whichever ID this belongs to, that's one of my parent cells. Once we have collected the parent cells and we now know the ID, we can now use this parent cell ID to go on to every other cell in the simulation, which is why this process might be a bit slow as there are too many cells to count. You're going to go to each another cell in the simulation and then check if that parent ID is contained in those progenitor cell lists. If yes, that means that that cell came from the same parents, and we're going to collect that information in this list called group. And that's all what this piece of code is doing. So it's going to the list of parents that you have identified so far. You're collecting the parent cell. And then what we're going to check for is that ID number that the parent has. We're going to look at each of the other cells' dictionaries. And if we find that parent cell in any of their information, we're going to understand that that cell is also a progenitor of the same parent cell. And we're going to collect that information. You might see this additional line here. This is another way of classification if you wanted to check if a cell basically came from the same progenitor, because a progenitor is always going to have a greater number of divisions than the cell that preceded below it. So I put this line in the code so that even if you wanted to sort of like modify any changes to the number of divisions, you can use this to sort of like classify cells even further. So this is the part of the code that you first need to copy, or if you're writing your own, make sure to include these lines. So found the unique list, fetch the parents, checked if that ID of the parents is in other cells list. If yes, that means that it's a descendant of that parent, and we're going to collect the parent and all its children into one unit called group. And the final part of this code is where we're going to color these groups. So for the group, once we know all the cell IDs that belong to it, this is where we're going to use that attribute which we labeled before, which is called color. So what we basically need to do now is we have information of how many cells and which cells make up that group, but we need to find out some sort of a way to represent them. One way to do so is basically by assigning them a number between 0 and 1, which is what we're going to do with your cell visualization field. So each group of cell is going to be colored with a color ID between 0 and 1. But the question is, what is the best way to color it? Because how many colors should we pick? So here I have done a simple scaling, which might look complicated, but it's really not. So all it does is we have a color index. It keeps updating. So if, for example, if you have 10 color groups, and now you want to distribute it from colors ranging from blue to say red, what is the best way to do it? You sort of divide the group number by the total number of colors. It will give you a value. That is the value that goes inside this field ID, and that is the color that this field ID is going to display. And that's pretty much what the lineage tracing is about. So even though it looked like, even though we started off with a simulation where we had a single cell, now using dictionary attributes and the cell visualization field, we're able to associate the field with a specific value of the color. Okay, I'm going to pause here for a moment, ask if there are any questions, and then we're actually going to try to run the simulation to see if what kind of outputs you're getting, and if we were able to fix some of the issues from before. So are there any questions with the general flow in which we are identifying cell lineages?
I just had a question, just clarifying. So like, so basically, it's taking its own, it has its own way of creating this color scheme. We're just deciding, we're just telling it, divide by this number of groups.
Yes.
And so if we wanted different colors for, it's like, because one thing I'm wondering, like, you know, they're all variations of like blue, green. And what if you like, and it can be difficult to see.
Yes.
Is it a way to change that? Or like, is it a way to like just more, maybe even if to change certain colors? I don't know.
Yeah. So, I mean, I think you're on the right track, because I don't know if there's a way to change the color map altogether in Compucell, which would make it more pleasing for certain types of shades. The way that we have done it is that suppose we have two different clusters of cell, then it's going to make the identification much easier, because you're going to have, say, 0 at very bluish, 1 at very reddish, and that distinction is easy to see. Now, as you fill in more and more clusters in between, the color map scale varying from, say, a number, if you have like 9 clusters versus 10 clusters, the color map is not going to change by that level to help you to see. So that's why there are definitely limitations to how we are assigning the values. But the other problem is that there are also only a finite number of colors that you can actually identify. And as your lineage keeps growing more and more, there is like we're going to run out of colors. So there might be ways probably we can make this even better. I don't know if, I don't think Compucell has a different map specification altogether. But yeah, we should probably, I mean, we should definitely think about ways we could reduce these differences between colors so that the nearby marked clusters are more visible to see, for sure. Yeah, as of now, I don't have a better solution for you, because as the smaller clusters, if you have 3 or 4, they're going to be more easier to see because they will be more spread out on the visualization map. As the number of clusters grow, it might be a little tricky.
Yeah, in the player, if you click on Tools and go to Fields tab, you can specify the maximum and minimum range of your values that you're using. So usually the maximum and minimum are set to be independent, to be non-fixed, but you can fix them to be from 0 to 0.5 or to be from 0 to 6, right? And then 6 will always be red and 0 always be blue. If you have that configuration on your player to have the maximum fixed, to be no, set the maximum field will be 10, then you can write color or assign a value to your scalar field to be something where in the range will be the color that you desire. It's a hack, is a way around. It's not. It's definitely not the best solution, but it's a way that you can actually have a little bit more control of the colors that appear on the visualization field. If I'm making any sense to you. Uh, Yeah, but definitely the best way to instead of just calling with a number, you call them with 3 numbers, where the first number is going to be the red, the green, and the blue. You just give this triplet of RGB numbers, and then the computer will do the color for you. That would be the ideal solution, because then you can do whatever you want.
Absolutely. Apart from, are people able to run the simulation and see the lineages with this code, or? Also, the reason I scaled down by the number of values is the following, so that we always sort of know the ranges over which we're displaying the colors. If you if you let the maximum and the minimum to be decided by the simulation in every case, you can do so, but then you would not understand how early or late these clusters were created. For example, when I look at the self-weight map, now I can sort of understand that the blue looking clusters were the first ones to get created, and the red looking clusters were the latest cells that have been created. If you let the maximum and the minimum to be unbound in a way, the colors are also going to the ranges are going to sort of like jump for each particular simulation. So if that's something you don't want, you can just rescale all the values as I've done here. But yeah, that's a limitation. If you have larger number of clusters, the range is going to get smaller and smaller. Are the simulations working for anyone so far? No. Okay. Let me know or stop me at any point. I'm going to move forward then to see if you would be interested in doing this exercise. So the idea is simple. So we have we have these dictionary attributes, and even we now have the information of each of these clusters with this attribute called group. So what we want to now do is sort of try to understand how many cells form part of each cluster. And this is a simple plot that I made by for this particular case where you have all of the cells uniformly growing. But you could also make this plot as you vary between the conditions, where you have, for example, picked only parent cells in between a particular region, picked only cells which have divided beyond a certain time, as might be relevant to real life lineage problems. And if you make a lineage tree out of that and try to find out how many clusters they have risen to, then these visualization plots are a helpful tool for understanding what sort of like cellular behaviors are actually changing the visualization output. So that is one of our goals for this, to sort of understand to add the scientific plot to visualize the size of the clusters. If you don't have, if you do not type in a simulation code so far, you can also download 5_3_4_3, and that should have the visualization code for you. So along with being able to identify the colors, identifying these lineage maps, you'd also be able to see a plot that tells you how many cells are in each cluster and what are the unique ID numbers that you obtain out of the simulation. So Google Drive downloads are not working. Is that a common problem? Or I can try to, let me see if I can. So I'm going to maybe just wait for a minute to hear back from you if you are doing this exercise. And if you have, if you have attempted this, or if you have any questions for, okay, thanks, Mike. If the simulation worked for you, and what are the sort of outputs that you're getting. So this is one of the exercises. The other exercise, which would be interesting to try, is, so we have implemented uniform growth. Oh, I'm sorry. This is the other instance I was talking about, which is basically selecting particular IDs in your simulation code. So in this case, we took the lineages from basically everything excluding that lineage number 1. But suppose now we say that we will only consider parents if they're within certain ID numbers. And then the other thing that would be fun to also try is, now that you can visualize these clusters, maybe you can ask also about the spatial positions of where these clusters are situated at. So because you have all the IDs of these cells, you can associate the cell objects to these IDs, and you can sort of plot the X and Y coordinates of where these clusters are being situated. So when you combine it with other features like non-uniform growth, for example, or only picking up certain lineages, do you see these clusters basically ending up in certain specific places? Or if not, how can you create lineages that only end up in specific places? So those are some of the questions that might be fun to try. And the other simulation that I'm going to pose as an exercise for the next 5 minutes, and then we can go over it at the end, is trying to sort of not think about uniform growth, but suppose I give you the problem that this is a situation where cells which are in contact with other cells don't want to grow anymore, and only the cells that are in contact with the outside medium are able to grow and divide. How would you sort of like demonstrate that? Just by looking at this lineage map, now you can guess that this map looks different than the one that we had before with uniform growth. So sort of, why do you get a map like this instead of the other one? And if, as you combine it with sort of the cluster analysis or any of the features, can you predict any interesting behavior to these lineage creation? So those are the sort of the exercises that I have for you. So I'm going to just pause and basically talk to you if you have any questions about all of this so far, and if there are any other exercises that you might like to try. But I think I can wait like another 5 minutes or so, and then we can go over the solution for this.
Yeah, I'm interested in this latter one. I'm not quite sure if I followed the question. I was like just
Okay.
listening to what the plot was.
Great. Okay, cool.
So, but I'm interested.
Yeah, so okay, I'm going to go back a few slides. So remember when we had this growth steppable, so we basically looped through all of these cells and we increased everyone's target volume uniformly by some target volume rate. So here we have not distinguished between any sort of growth or division properties. We said that if your cell present in a simulation, just keep dividing and growing and dividing. But we could also ask the question, what happens if not all these cells are allowed to divide? What if cells only at the periphery are allowed to divide? Only cells at the periphery of the simulation. By periphery, I mean something that is still in contact with the medium or the external simulation space. That is an interesting thing to think about, because people have actually shown that cells when you culture cells in media, you have something called contact inhibition of proliferation, which means that cells which are surrounded by other cells can actually slow down or decrease their growth rates, either due to mechanical constraints or due to chemical factors as well. So the question is, if we now go from a uniform growth rate, how would we model something that is non-uniform? And what would we change in this code to only have cells that are at the periphery of the simulation that can only basically grow or divide? In that case, I've already shown you the answer, that the simulation map that I obtain is something like this. And maybe you can tell me why this is the sort of map that emerges from this simulation, unlike the other simulation. And how would you actually implement this from the Python information you have?
Well, I'm thinking like from the gradient from the other plot, since it was linear, it was just more able to add. You can add from the edge of well, I know a circle doesn't have an edge, but it can be concentrated. You can kind of see like a trend where there's more colors. I'm trying to explain this basically. Like if a circle were to have an edge, you could see the concentrations, because you'll see more color added into like a circular, added into the circle. Let me say it like this. And then with this contact inhibition, the growth will, I wouldn't have imagined it would look like this, but it would be much, because if contact inhibition kind of shows that like cells, they're going to like move in clusters. So they would develop in clusters versus in a linear. And so you have this clustering pattern, because like all the yellow ones kind of move together, all the green ones kind of move together. And I guess so the blue one would be like the parent, but so that's why it's like in the middle, and the other ones just kind of spawned from it. But that's why you have these clusters, because when they are touching each other, they'll kind of come in waves versus a more sequential pattern. That's the only way I can explain.
No, I think you're absolutely on the right track. So what happens is, so the contact inhibition affects all of these cells. similarly. So in the other case, what was happening is this cell is still able to divide. So it's able to produce lineages that sort of make these like circular traces that you can see. Of course, these are some of the earliest lineages because they have the maximum number of cells because they divided so much earlier. In the contact inhibition case, what happens is that this guy is not able to divide anymore. So the cells, once it creates these initial number of progenies, the moment it gets surrounded by these cells, it has no way of basically proliferating anymore. And then these lineages, which are already created, they have their own, they have already surrounded this blue cell, sorry, and then they keep on dividing. And this division, they sort of, it seems like they have like radially protruded out, but it's only because there are no more new lineages coming in. All of these cells inside have no way to proliferate. And the only, because these divisions are also localized, they're right next to the whatever cells they have created. It looks like they have like radially emerged out. But it's what's adorable about this is that you have maybe like 2 parameters that you changed. One in the case of uniform growth versus now where you have a more contact inhibited growth, and then you're able to change the cell fate map in a more in a different way. So I think these, even if you played around with the growth division codes and try to come up with their own rules of saying, hey, I'm only going to divide if I am at a certain distance from the initial stem cell, or if I'm at a certain distance in the lattice. And that could create a whole new set of fate maps that you can see. I hope that, yeah, but you were absolutely on the right track. And yeah, it's, so did the simulations work or not work? Even trying. So I'm going to just post the solution of how did the contact inhibition. So basically, instead of the uniform growth, what we now can do is we can go to each of our cell objects, and what we can additionally find out is the neighbor of each of these cells. And once we know the neighbor of each of these cells, if we make the classification that it can only divide if it's a medium cell, then we have a contact inhibition of growth, a very simple model. I think Gilberto probably talked to you about a more sophisticated model where you can actually also measure the pressure pressure for each cell, and that pressure could dictate whether you divide or not. And those sort of models would also be very interesting to see in the context of these visualization fields. Just to clarify what this piece of syntax means, if you're wondering. So when Compucell generates neighbor lists, every cell that is a cell type will be called a neighbor. It does not classify medium as a neighbor. So that's why it means that if you're not a neighbor, which means you are not a cell, then that means that that you're in contact with some sort of medium. This is where you start incrementing your volume. So in this simulation, the cells that are basically growing are only at the periphery of the simulation. And this is another one of CC3D's funny syntaxes.
Wait, I'm trying to make sure I'm following. So you're saying,
oh, sorry?
I'm trying to recapture. I'm trying to like take in what you just said because
I can explain it again.
Yes, if you could.
So does it make sense of what this list is doing? So for each of these cells that we're parsing through, we're creating some sort of a neighbor list by parsing through the neighbors that surround that particular cell. And all of these neighbors are going to be part of this neighbor list for each cell. If it's not a cell, and it's still so if you have entered the neighbor list, but you are not a neighbor, that's Compucell's way of talking about a medium cell, basically because it's not technically a cell. So it does not have a particular cell type. So if you were, for example, looking at another neighbor cell, the syntax would say if neighbor. But if you don't want it to be a cell type, the syntax is if not neighbor. That means if you are medium, then you can start to expand.
So basically because we've only had, in this case, we only had medium and cell, is basically essentially saying anything that's not a cell.
Yes, exactly, exactly.
And
Yeah,
so
yeah,
if we had two cell types in the
Mhm.
medium,
Yeah.
then if one of those cells were able to proliferate, we would have to just specify, or
Exactly,
again,
you
if
got
we
it.
just,
Yeah,
or if we just wanted one, it would exclude the medium and the cell type that we don't want. Okay, I got you now.
exactly. You got it. Yeah.
We need a lot to expand. Okay.
Yeah, sorry, this is weird Compucell syntax, but yeah, that's how you reference to medium using a neighbor list.
Can I expand on that question a little bit? As you're looping through that inner loop, and it's giving you those values of neighbor and common surface area, would it be fair to say that if you have neighbors that are cells, the value of neighbor is the ID of that cell?
Absolutely. You could do something like if neighbor.id equal to 1 or whatever.
I see. In that case, I think I can explain the syntax here because the cell ID of the medium is always 0. So neighbor, it's saying if it's looking at neighbor and it's saying it, if not neighbor means if neighbor is 0. The 0 is interpreted as false.
Absolutely, you got it. Yeah.
Let me interject here. Is almost like that. It's just that the neighbor is a known object. It's not defined. So if you do if neighbor with something that's actually not defined, you return false. So to get the ID of something that is a cell or is a cell object, you have to give the cell.id or neighbor.id. Then you return the number.
Okay, perfect. So that's a reference to the cell.
Yeah. So if you have the neighbor, if the neighbor is a medium, if you try to do neighbor.id, you get an error because the neighbor is not an object.
Yeah. Got it.
But the idea is the same. Yes.
Yeah. That's
Just
even better.
details.
That makes more sense. Thank you.
All right.
So this is what I have for you. Are people still doing exercises or do you want more time? Are you running any simulations?
I'm interested in, I think you had the second one. I don't know if we had, it was, what, I think it was like, I can't remember what the activity was. It was, you were presenting 3 of the exercises. And then this one. So I'm interested in this. So this one is building off of that, like, because I was typing in the code as we were going. So if we had the code, this plot or using this plot, I'm interested in trying to, is it telling different information than the other plots? Like, is it?
So if you are running the simulation, then there's already a plot code embedded into the simulation. The idea was to sort of gain practice by making scientific plots and collecting information from the code to put it in the scientific plot info and sort of like display what it wants. So the idea for this simulation was that if we have we were collecting objects called groups, which is the collection of all the cells that make up a particular lineage. And so one of the tasks we could do is sort of ask how many cells form part of each group. And how would we basically plot that information? And a way to plot it is Compucell has functionalities of generating these plots. They're available in the CC3D Python if you go to scientific plot, and they have different levels of where you can first set up in the start function a scientific plot, and then this will be the in the setup function. What you're basically doing is setting up this window where you'd be putting in your adding in any data curves that you want. And in the step or the start function is where you'd be adding any additional data information. So if you set up this plot in the setup in the start function, and then you move over to the step function where we were collecting all of the cluster information to basically construct our lineages, what you could do is sort of collect the information from group on how many cells form part of the cluster. And you also know the parent ID that sort of initiates or makes up these groups. And you can plot on the x-axis, the ID, let's say, and you can plot the number of cells on the y-axis. So for example, this plot is very simple because it shows you that the ID of 2 had the maximum number of cells, which makes sense because since it was created at the initial beginning, it had more time to perform a bunch of divisions. And so it's going to end up with a more number of cells. So that's why what you see is that the ID which was created at the end has fewer number of cells. So that way the plot is very easy to interpret, but it still gives you some sort of practice with how to basically set up the scientific plot and sort of generate or combine data from inside a simulation that's running to be displayed on a scientific on a plot, basically. Couple of things that usually happens when you make these plots is unless you erase the data at each time step, the data is just going to get superimposed on this, so it's harder to understand. So some recommendations are, if you're calling a scientific plot, set it up in the start function. When you go to your step function, make sure you erase the plot before every run or whichever runs concern you so that you have fresh data to look at. And then you can add the function of add data to whatever plot window that you created, and those attributes of the plot are going to be information that come from your code. In this case, I chose ID for my x-axis and the cluster number for my y-axis, but you could easily shift it and, for example, plot the number of divisions or the number of cells as a function of Monte Carlo steps and see how they increase and what sort of growth curve you would expect from a sort of a uniform division. So you can do all sorts of plotting with these cells. This is just from my example simulation. You could also combine this plot with the contact inhibition case that you just saw, and sort of ask if any of the if the diagram changes in any way. You could also only extract out cells within a particular positions, and then try to understand how many cells make up part of that lineage as well. So the idea was to get you to practice a few of these scientific plots and use whatever variable from the code that you like to visualize it on a graph. Thanks, Rosie.
Yeah, this stuff is really helpful. I was running, sorry, I'm running the code that was uploaded, and I think my Nano Hub is just crashing or going really slow.
Oh no.
And so, but yeah, it's very helpful. So I didn't realize you can specify kind of exactly what you want to look at as far as
Yeah,
you
so
can do a lot of things with these plots essentially. Because I guess I'm thinking if you have like so many different cells, like if you're looking at hundreds of cells, then you can use something like this to get a high level view. And if you wanted to look in on like
yeah,
earlier lineages, I might not know, but the simulation will be able to identify like all of the cells that were a part of a
yeah,
certain lineage. Okay.
absolutely. You could plot something like the lineage ID with respect to the X position or the Y position. And so it's going to be like a spatial map where it knows what the coordinate is, and compared to either the X or the Y position, or if you can plot both as a function of some color, so you can see where they ended up. The visualization field do a good job of telling you what the spatial variations are, but this is another way of being able to compress all that information and put it on a like graphical nature. Yeah, and the scientific plots are, so these are all internal to Compucell. Other ways of which you can do is you can also use an option called print. So the print command in Python lets you print to a file if you need to be. So if you have, for example, data that you can say print to a particular file and you label the data columns that you want, it will basically generate these files for you, which you can use for plotting in any other software too that you want. So these scientific plots are for internal Python viewing, and you also have options of like importing the data to plot elsewhere.
Okay. I have a question. Have you ever used this plotting? I'm very interested in like how you like condensing a lot of like data information. Have you ever used this for like several thousands or millions of cells, or like omic type of data, or just like big data? Or is this yeah, can it be used for like just large amounts of like very large amounts of data,
So
or will it break?
so it depends on the thing that you want to plot mostly. So for example, Compucell is, I mean, if you have 1,000 cells, Compucell is not going to do a good job of running the simulation at a good pace. And because this is like internal to Compucell, so whatever data Compucell is producing for you, this is a good way to visualize that data. Suppose you have some sort of external data that you have on the scale of a large amount of information, then something external that you do not need like Compucell to process or generate a model for you, then it's best to use something external. But if you are running a simulation with with a lot of cells, then you can, I mean, yeah, depending on what is the output you need, you can you can plot, you can decide the variables that you want to plot. Yeah, as the simulation gets large, Compucell is going to be very what do I say, like upset about it. It will take a long time to do large. It takes a long time to do large simulations, the scale of like thousands of cells.
Okay, that's good to know. Thank you.
But do you, if you want, we could try. I mean, it's up to you. So if you want to try out running a scientific plot on any steppable function, let me know if I can do anything to help. So it's all of this information is in the CC3D Python, and then yeah, scientific plots, and then you'll have to do first you'll need to like set it up so that you have a window that's going to basically identify with the plot that you're creating. So there will be the option of creating a main window and giving it a main name. Then part of that main plot is going to be these graph or lines, whatever you're creating. And that information from the lines can be either part of the step function, or if you have a data point in the start function as well, you can add that in.
Is there like best practices of one versus the other, like if it should be in the start, or does it really matter?
Right. So suppose you have a quantity that's changing in time. So then it makes sense that your plot is also keeping track of this quantity and evolving with it. if you have a quantity that you needed plot at the beginning of the simulation, and you just wanted to see how the features look, and you just want